So I think I had done this for innate where I had divided up the lectures. I know I'm definitely probably going to have 2 parts, so this is just the first part of adaptive. And we're actually going to do an activity one day next week. So that should be fun. Get us doing something and get you guys talking instead of just me all the time. It's fun, trust me. I did it with my other class and they really enjoyed it. And they're like, we should do this every day. And I was like, no, not possible. Yeah, if you talk about it, which is why, it's important sometimes and I make the time to do it. So we've talked about innate, what you're born with, right? And although these 2 systems aren't separate, right, we kind of talk about them separately, but they're intertwined. So adaptive is what's going to adapt and change over your lifetime, okay? So it is the most effective means for eliminating invaders. The drawback to this one, though, is that it could take a week or more to develop that very first time you see that specific antigen, and I mean specific, right down to the molecular level. That's why this system is so effective. I should have just done it ahead of time. So your innate, right, this not very specific, right, what you're born with protection has to protect you in that meantime, in that week or longer that your adaptive is coming online and recognizing the first time an invader. So the unfortunate thing is sometimes we don't survive, right? That the pathogen overwhelms us and kills us before our adaptive comes online. Right? This is true for things like Ebola, right? Most people, their immune system doesn't kick in soon enough and they would be killed by that infection. That's why it's so scary, right? It's a very aggressive virus infection. The good news and why we're trying to develop vaccines is the second time you see something, right? Once your immune system has seen it once, it remembers it, right? This component of your immune system has memory, right? So the second time it responds much faster, or the third time or the fourth time. So vaccination takes advantage of this memory component. And what we're hopefully doing with vaccination is it's your first exposure to something not as damaging as the real thing, something that's been weakened, right? So even pieces of a bacteria instead of the whole bacteria, or a weakened bacteria, or a killed bacteria, or modified viruses. As I said, it's specificity right down to the molecular level. So I've talked about a particular strain of E. coli before, right? E. coli 0157. There is specific sugars on its surface to denote that particular strain. And the bad news with that strain is it produces really awful toxin called the sugar toxin. So we can detect it with antibodies, right, with an immune response all the way down to that very specific difference between that strain of bacteria. So it's right down to the molecular level, to the sugars associated with that strain of bacteria. Then also this system must be able to determine between self and non-self, and really between healthy self and non-healthy self. So this system can even eliminate cancer cells, right? Cells that are behaving badly, if it recognizes that they are unhealthy, even though they're self. And of course, pathogens we want to denote as not being self, as being dangerous. So these are our cute little cartoon guys that I took from the first website that I sent you guys to as an introduction to immunology. And so he says, I recognize this one. So literally down to the cellular level, this is a lymphocyte. He's specifically recognizing that specific pathogen, whether it's a bacteria or a virus. And then this guy over here, again, is showing that specificity, but a little bit more detail where he's got a receptor, right? And they actually bind with the antigen, right, with this foreign substance, with this bacteria. But again, it's at a small region. It's very molecular in interaction. So these little crowns that we'll give a more specific name to later are a specific molecular complex on the surface of that cell that the receptor is actually binding with. So there's two types of adaptive immunity. There's what we refer to as humoral, which is a lot of times just nicknamed antibody immunity, because that's the most common product of that stem of our adaptive immune response. And cellular is, again, a cellular response, and this is for when stuff is inside of our cells or when our cells themselves are behaving badly. So we can detect outside of our cells, that's our antibody response, or inside our cells is our cellular response. So we have ways to defend against just about anywhere it can get, whether it stays outside of our cells or just outside for a short period of time, or if it gets inside of our cells. So I'm going to play this general animation, right? So this is kind of an overview of the stuff we're going to be covering in the next couple of lectures. And then as we go, we're going to be adding in layers of detail.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So as I said, this one's a good overview, right? There's only a couple of things that upset me, and one is somebody messed up in their animation, as we'll see later. It's interacting with the wrong receptor here. It's supposed to be interacting with this purple one over here. So, yeah. Yeah. And I didn't even notice for the longest time, you know, because they show it relatively quickly, but that it's actually binding to the wrong spot on that cell. And another thing that they don't mention, they show the memory B cells, but whenever a T cell becomes activated, it also is going to produce memory cells. So in overview, the first exposure, the very first time your immune system sees it, we refer to that as the primary response. And as we said, the good thing about this is memory cells are going to be produced, okay? So we have the ability to remember this. And so the second time we encounter it, it's a much faster, swifter, stronger response. And any subsequent time after that. And then, as I said, the humoral response, sometimes referred to as antibody response, is specific against extracellular antigens. And we're not saying necessarily ones that stay outside of your cells, but it can defend against anything that's outside of our cells. So viruses, although they get into our cells, there's a time when they're outside of our cells, when they might be in the blood, might be in the tissue fluids. Antibodies can be protective in that manner, right? Bacteria, toxins. Again, at some point they may enter into our cells, but there's a time frame when they are not inside of our cells, right? So when we say extracellular, we're referring to that the action, right, of this type of response is extracellular. It's outside of our cells. Antibodies cannot go inside of our cells. Make sense to you guys? Okay. Where cellular immunity deals with antigens within our cells. So definitely when viruses are inside of our cells, we want to be able to detect that. Even some bacteria, some protozoa, different pathogens actually get inside of our cells. And we want to be able to detect this, right? But that being inside of a cell is an added level of difficulty in being able to detect. So we have ways of detecting both those fronts, whether it's outside of our cells, extracellular, or whether it's cellular inside of our cells. And different white blood cells are going to be involved in these different modes of detection and defense. So both of them are very powerful too and can damage our host body defenses. Towards the end of this chapter, we talk about what I refer to as disorders, where the immune system is attacking self with autoimmune disease, or some of the hypersensitivities, or what we commonly refer to as allergy. Okay, although you'll find out this is a normal progression of immunological response. The problem is what we're responding to usually isn't something that's going to harm us, isn't a pathogen. Granted, it's foreign, right? So the body is treating it as such. Unfortunately, it's not something that'll kill us. And then as you saw, that they actually are interacting with each other, right? So there's receptors that we'll look at and that we'll name, where they're literally interacting receptor to receptor with each other. But in addition, they release substances like interleukin 1 and 2. What's the general name we give to those molecules? Those chemical signals, where they're like talking to each other. Their voices, but what's the scientific name for their voices? Cytokines. And interleukin's a particular class that tells you who's communicating here. If it's inter, it's in between. Leukin refers to leukocytes, so white blood cells, right? So the white blood cells are talking to each other, right? Those were all white blood cells that we were looking at, communicating with each other. You don't need to know the specific, like what IL-1 and IL-2 do, right? But you do need to understand what cytokines are, right? That they're the chemical message, and that particular class is specific to talking between white blood cells. And there's a lot of them. So we're going to stick it just to those few classes, right, and that level of understanding. So this is an excellent schematic, kind of gives you an overview of the adaptive immune response. So remember this cute guy, our dendritic cell. Although he phagocytoses, one of his major contributors in his functionality is presenting this information to the T cells, right? So notice two different types of T cells that we're going to talk about, cytotoxic and helper T cells. The cytotoxic ones will actually kill cells, right? So this is what we refer to as our cellular arm of the adaptive immune response, dictated by cytotoxic cells that will actually destroy infected cells, our own cells that are infected. Helper T cells, on the other hand, do just that. As the name implies, they help when they become activated, right? And so they can help out macrophages, increase in their killing ability, right? Because they're one of our main phagocytic cells. And then they can help out the B cells. And as we saw in the animation, they're going to go from B cells into plasma cells, right? And those are going to actually secrete antibodies that can directly interact with the antigen. Antibodies are proteins. These are things that are made by the plasma cells and secreted. Now, this step isn't always needed. You don't always need the helper T cells to activate the naive B cells. Sometimes it can be done on its own. So how do they interact with antigen? So these B cells and T cells, right, they're right at the top of being coming in contact with antigen. Now you saw that the B cell actually has a pretty unique B cell receptor. It's actually an antibody, and we're going to go over all the different types of antibodies and their structural components today. It can bind with what we call free antigen all by itself. And these antigen binding sites are the same. So it could bind also with another antigen. Unlike the T cells, which have just one antigen binding site on their T cell receptor, and T cells cannot interact with antigen all by itself. It has to be presented by an antigen presenting cell, right? So one of the examples that we saw in the animation was a macrophage, right? It can gobble it up, but then it can present that information to the T cells. Dendritic cells, as you saw in the drawing, they can present, okay? B cells themselves can present. Another thing that happened in the animation, unfortunately, the wrong receptors were binding. Okay. And then any of our cells that get infected have the ability to present, okay? So our specialized cells that specialize in antigen presentation have a specific receptor that's going to grab onto antigens inside of them or that they've engulfed, right? And so this is called MHC. We're going to define it out later. It stands for major histocompatibility complex because this molecule is the one that creates problems when you try to do tissue and organ transplants, because this is the molecule that our T cells are not recognizing and create a problem. So I have an animation that I'll put up for you guys later and that we'll watch that shows T cell development and how important it is that the T cell actually recognize MHC, our MHC, that we use to present antigens, right? So this very important cell-to-cell communication, I almost consider it like a special handshake, because there's a particular marker on T cells. They're called CD markers, and the two different types of T cells have different markers and interact with different MHCs, right? So it's a specific interaction between these cells. They only talk to certain cells. So now for our antigens. We've just been using the general term antigen, right? Which means antibody generators. But we go down to the molecular level in these interactions in our adaptive immune response. The B cells binding with their receptor, which is an antibody, the T cells binding with their T cell receptors. This is down to the molecular level. So where that binding is actually happening on the antigen is referred to as an antigenic determinant or epitope. And I'll always refer to them as epitopes, but you should be aware of the other name, antigenic determinants. So as we said, the name antibody, antigen actually, comes from the root of antibody generators, because this was one of the first products of the immune system we could easily study and understand. Today, sometimes people will use the term immunogen, right, instead of antigen, to refer to the fact that antibodies aren't the only thing we make in response to an antigen, right? It was just one of the first things we discovered. We have T cells, right? We have a cellular response other than the humoral antibody response. So, and in general, even against viruses that get inside of our cells, we'll respond both ways. We'll produce antibodies and cytotoxic T cells. So we can get them when they're outside of our cells with antibodies, and we can target them when they're inside of our cells with those cytotoxic T cells, right? So we're going to come at them any possible way we can. So an antigen, right, or an immunogen is really just anything foreign that's going to elicit an immune response. Proteins and polysaccharides, because sometimes they're repetitive, can induce what's called string response, and we'll look at a picture of this later, where lots of the antigen can bind to lots of the receptors on the surface of the B cell. This can help initiate the B cell to move forward to producing the plasma cells and secreting antibodies without T cell help. Lipids, nucleic acids, don't tend to be very good antigens. and definitely don't cause the type of string response. So we usually need some type of confirmation from the helper T cells for the B cells to move forward. And so the next animation we're going to look at goes over that specificity that I was talking about, right down to the epitope.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So as I said, it's molecularly specific. And this response here, where you see more than one different antibody against a particular antigen, a very large substance, even a protein, right, as you saw, multiple different antibodies produce binding to different epitopes on that actual protein. This is referred to as a polyclonal response, and this is typically what your body does. Poly meaning many, clonal referring that each one of the B cells will clone themselves and they produce a specific antibody. So, you know, all the yellow or orange ones denoted here were made by a particular set of B cells that were all the same, right? Because each cell is programmed to make a specific antibody, right? And again, it specifically binds to a specific antigen right down to those epitopes, those little pieces. So we're talking about as little as, say, 10 amino acids or so that the protein, that antibody is actually binding to. Now we look at antibodies, but T cell receptors are the same way, right? Where they're just binding to a little portion of the antigen that's being presented by the cell. So proteins, as I said, generally listed really strong response. Lipids, not so much. They're highly repetitive with not much variability, usually for lipids, which is why they don't make good antigens. Very small molecules are usually not antigenic, which means they're just not big enough for the antibody to bind to, okay, or to elicit an immune response. And then, as you can see in this bacterial picture, they actually have ones binding to the surface of the cell and others binding to the little protein appendages, the little fimbriae that are coming off from that cell. So anything that the antibody can bind to, it will, right? But it's a specific interaction that happens. So how does our body make antibodies? Okay, so each one of our B cells, I almost equate this to playing the lottery. Picks from its genetic information. There's millions of different antigens out there, and they're always changing, right? Viruses and bacteria, they're never staying the same. Their DNA is changing too. It's mutating, it's changing, it's being selected for. Our DNA, there's no way we could have enough information when we're born to make all the different antibodies for all the different antigens out there and then keep up with the ability of change. So what we do is we manipulate our DNA, right? We have segments in our DNA, and so like I said, similar to picking the lottery, you have a set number of genes, right, segments of DNA that we're going to pick from, and we're going to combine them together. So that's referred to as gene rearrangement. They literally rearrange, cut out pieces, and stick pieces back together, right? It's a permanent change for these guys. And then when they're doing that, and the animation doesn't show this really well, when they join together the different pieces, they don't precisely join them, right? So I can't think of an analogy that would work. This morning I talked about kind of like a jigsaw puzzle. You know how those pieces fit together perfectly? In this case, when they pull apart these pieces and put them back together, it's not a perfect fit. And they don't always cut the segments in the same places. Right, so we get variability in the DNA code by imprecisely cutting apart and joining these pieces together. Does that make sense to you guys? Maybe a little bit.
Is there a name for that?
It's called imprecise joining.
Oh.
It's just imprecise in how they're putting the pieces together, right? Rearrangement is where they're actually literally picking the pieces, and you'll see that really well in the animation. And then when we talk about combinational, there is 2 sets of genes that are going to be rearranged to form this antigen binding site. So that combination of those 2 together helps in the variability that creates the diversity of all the different antibodies. So let's watch this animation and see if hopefully clears up how this process happens for our B cells. Now, again, this is B cells and antibodies, right? The same thing happens for our T cells, because each one of those guys have a specific antigen-specific receptor. Theirs isn't quite as complicated as antibodies, though. That's why we choose to go over antibodies.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chains of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
So as they say here, this is B cell number 1. B cell number 1, this is their lottery numbers they picked, and they've literally permanently rearranged their DNA so that they're only using V3, D1, and J2 for the heavy chain, and V2 and J1. And even then, if some other B cell happened to pick this same combination of light and heavy chains, they would still be different because of that imprecise joining, right? Because they don't perfectly cut out all of what's designated maybe D1, right? They may take only part of it, right? Still with me? So then another B cell, when it's being born in our bone marrow, right, is assembling its antibody, and it happened to pick, say, these combinations. Now the reason why the heavy chains, and notice they're longer, hence why they're heavier, and these are protein chains, right? Antibodies are proteins. And the light chains, of course, are lighter because they're smaller. They come together, so both sides of this molecule is identical, okay? They're both the same. Down here, this region, this constant region, is constant for each one of the classes of antibodies. So as you guys can guess, how many different classes of antibodies are there? 5, right? Because there are 5 listed here, right? And the different ones interact and have different functions in our body, and interact with our cells and our immune system in different ways. And so we'll learn about the different classes today. So as I said, right, even though we watched B cell, T cells go through this same process. And they're going to be checked out to make sure they do proper functionality. So this process is a little bit more involved for T cells and happens in the thymus. And this is actually where they get their name T cell, right? Because even though they're born in the bone marrow, they must travel to the thymus in order to make sure that they interact with MHC properly and that they're not recognizing healthy self-antigens, right? We don't want T cells that recognize, because this is a random process, right, of producing these B cell receptors, that antibody on its surface, and the T cell receptor. It's a random process, like picking your lottery numbers. So we might pick a combination that interacts with our own self-antigens. If that happens, we don't want those cells getting out, right? Because then they would attack our cells. So that process all of that process happens in the bone marrow for B cells. For T cells, a large majority of it is going to be done in the thymus, right? Where they're recognizing the receptors they need to recognize and making sure they don't recognize healthy self-antigens. So the next thing we're going to look at is the antibodies themselves. We're going to look at the 5 different classes. We're going to look at what these antibodies can do, right? Once that antibody binds, why is that beneficial, right? And one of the things you guys saw in the animation is a phagocytic cell could grab onto that antibody, right, and ingest. And we call that tagging for destruction. There's actually a specific name we give for it. It's opsonization, right? And we've used this term before, right, with complement, which we'll review a little bit today, right, in that process of recognizing. So as you can see, there are already parts to the antibody, right? There's the heavy chains and the light chains. They're what we call these variable regions and these constant regions. Where are they and what are their importance? So this is one of the ways that I could put a picture on the test, right? And I could label each part with letters and ask you to denote which ones are which. So functionality-wise, what's really important about these guys is their ability to bind antigen. So the antigen binding sites are at the top here, right, of the light and heavy chains where that variable regions they talked about come together. And both sides are the same, right, and they're both going to bind to, say, the same shaped antigen at both sites A and B. C down here denotes this portion of the antibody, and this interacts with our immune system, whether it be cells of our immune system like a phagocytic cell or components of our immune system like complement, right? This is the part that the immune system is going to interact with. This portion is what's actually going to bind to the antigen. And in order to do that, we have to have this structure. And so we've already seen we make 2 identical light chains, we make 2 identical heavy chains, and they come together to form this characteristic Y structure. Right? It's a stick figure, as you will, for an antibody. They're actually globular-like proteins, but they have an overall Y shape. It's just not quite as linear as shown here. Right? When scientists were studying antibodies, they could break them up into those 4 pieces, those 2 heavy chains and those 2 light chains. But they also found that they could break it right here at the hinge, and they would end up with 4 pieces. This part A, B, and C. And what happened with the C fraction is it crystallized. So that's where it got its name FC region.
Wait, did you say 4?
No, 3 pieces. No, when this one, let me back up. So here you would end up with 4 pieces if you break up the chains. You got 2 heavy chains and 2 light chains, right? So they were able to break it up into those 4 pieces, right? 2 heavy chains and 2 light chains. That's where they get their names. The heavy chains, as you can see, are longer, therefore they were heavier, and the light chains are shorter, so therefore they were lighter. They are. Shouldn't say were. They are. Now, when you break it here at this hinge region, you get this piece here, A and B. And notice these are the same. They're just mirror images of each other. And what they do is bind to the antigen, right? Remember the antigen binds up here? So they're called the AB, the FAB, for fraction antigen binding regions. right? Fab. But it actually stands for antigen binding. The C, on the other hand, as I said, happened to crystallize. So that's where it got the C designation. It wasn't actually because they were doing ABC, alphabetical order. But it kind of works out that way. But this is the portion that, as I said, our immune system is going to interact with. So we'll use that abbreviation FC a lot. And that's what I'm talking about. I'm talking about this portion of the antibody, this region of the heavy chain.
Does it crystallize because of the light chain?
I don't know. She asked why it crystallizes. I have no idea why it crystallized when they separated the pieces. Good question. Never thought to ask it, though. So why do we have this overall structure? Okay. Part of the reason is because of these constant regions and variable region. Variable we need, right? We need variability for us to be able to, for each B cell to be unique, right? And have a specific receptor for a specific antigen out there. Don't even know which one, right? They just pick their numbers and hope they're going to win. So both the light chain and the heavy chain, as we saw, produce a variable region, right? That's going to vary from antibody to antibody. So denoted in the green here on this picture, on this diagram. Now, for the light chains, they have a short segment of constant region. And this constant region has to exist, has to be continuously the same from antibody to antibody. Otherwise, the light chain will not bind with the heavy chain. And we won't get this overall Y structure. Same thing is true for the two heavy chains, right? This is all constant region so that they can bind with each other. And there's flexibility in that region. That's why it's kind of called the hinge. It's not stiff. And then this portion, right, so this, all this blue is constant, right? But when we get to this portion down here, what we call the FC region, will vary from class of antibody to antibody, as we saw, right? Encoded in our DNA, we have 5 choices. We can do IgA, and when we say Ig, we're saying immunoglobulin, right? That's what Ig is for short, which is the same thing as saying an antibody, right? So whenever we say Ig, we mean antibody, okay? When we say the next letter after Ig, whether we say A, E, M, G, or D, we're referring to the specific class of that antibody. And as I said, different classes do different things, right? Interact with the immune system in different ways. So here, right, our antigen binding sites would be M and N, and remember the O is denoting down here, this region here is where the immune system is going to interact. If we were to look at the FAB fractions, right, so the fraction antigen binding regions, notice it would be A, B, C, D, E, F, H, G, and H, right? Both these top portions. And then constant regions, remember they start further up, right? So the second region in both the heavy and light chains is constant. And then down on, this is considered constant region as well for the heavy chains. Here's a bacteria, right? Here's antibodies attaching to it. And again, the blue is denoting the variability, right, of this particular antibody, where the red is denoting the constant region. Now, that FC region, this bottom portion, you can almost, sometimes people call it the stem, right, and sometimes call these the arms of the antibody. There are some white blood cells, okay, that have receptors on them for this FC portion. And in this case, it's even to a specific class of antibody. It's IgE. So IgEs can bind to the FC receptor on mast cells. And who's the close cousin to mast cells? Mast cells are in your tissues. Who'd be found in your blood? What's the fill that's similar to a mast cell? Basophils. Yeah, basophils. So these particular antibodies interact with these particular cells, mast cells and basophils. And again, it's because this portion, this FC portion, they're recognizing, right? and right down to that specificity even of that class of antibody. Because each class, that stem region, that FC region, is going to be different. So as I said, that Y shape can be broken down, as we saw, into those 3 fractions, right? And so 2 of them are identical. Those are the antigen binding, right? The 2 arms, sometimes referred to, and the stem, right? Very important in that it is the FC region, right? This is going to denote what class of antibody you're looking at, and this is where the immune system is going to actually interact, right? So as you saw, that mast cell binding to that portion of the antibody. And again, the C comes from crystallization, but I always just remember constant, or class even, right? Because again, it's the different classes of the antibodies that's determined in that region, and it's pretty consistent between the classes as to that molecular structure. So here are our 5 classes. In general, most of them are in this monomer form that we just looked at, right, with the 2 heavy chains and the 2 light chains. Some of them, when they're secreted by the plasma cell, or in the case of IgA, it's actually secreted by our body in our secretions. I'm going to give me an example of some of the secretions that help protect our body, part of our innate immune defenses. No, I'm talking about not the actual chemicals, but the substance. So mucus, right? What else? Tears, saliva, right? All of these, and then for women, we've got one extra secretion. It's called breast milk, right? So in these secretions is IgA. In this form, this dimer form, right, where 2 monomers are hooked together. So notice, right, and again, there's an extra molecule that's hooking them together. So notice their 2 FC portions are pointed towards each other. That's because, again, it's leaving our body, right? Tears is leaving your body. Mucus leaves your body. Even though it's all lining what we consider the inside, it's actually your outside, right? Your digestive tract is actually considered an external environment, okay? So breast milk, definitely leaving the body, right? Saliva, again, is part of the digestive tract. And so, again, considered an external environment, right? We are secreting it. So there are no cells that are no white blood cells that are going to interact with this. Right? They don't have to have access to that FC portion. Complement's not going to be there. It can't interact, right? No components of our immune system are actually going to interact with these guys. Well, that seems kind of bizarre, huh? We'll talk about what they do. They do protect us, right? We're not just throwing them out there just to put them out there. They are protective, but they have a specific type of protection they can do. So IgM is the class of antibody found on the surface of your B cell as that B cell receptor, right? So when it makes it, it puts it on its surface, it's its receptor for antigen. But then when cells turn into plasma cells, they can secrete, right? And in the case of IgM, when they secrete IgM, they're going to put it together in this huge pentamer form. And so I always remember IgM mega big. Right? This baby is huge. It is 5 monomers hooked together, 10 antigen binding sites total. This guy can bind some serious antigen. IgG is always going to be in this monomer form, and IgA, when it's in your blood, right, it's in the monomer form, okay? But it's very little is in your blood. It's just traveling if it's there. Most of it is produced along your mucous membranes. It's going to go right into the mucus. And you produce a lot of IgA. IgD, we actually don't know much about it, and we've actually found it on the surface of the B cell as well. But the primary class is IgM. So there's still a lot we don't know about this one, so I'm not going to have much to say about it when we get to the detailed slides. IgE, again, in this monomer form. And this is the one related to allergy. So how I remember this one is my first name, E for Erica. Erica hates this antibody because it deals with allergy. Something I struggle with all the time. I have way too many of these IgEs. Wish my body would stop making them. So for IgM, right, it's on the surface of our B cell as that monomer, right, the B cell receptor. But when we secrete it, it's going to be in this pentamer form, this mega big. And when it's in this form, circulating in our blood, it doesn't leave our blood. It's too big, right? It's a really large antibody. It is the only antibody that the fetus produces. Right, so they do produce antibodies in utero, but just of this class. And this is the first class you're going to make most times. T-independent antigens, meaning that they don't usually, these types of antigens, to get the B cell to produce IgM, you don't need help from the helper T cells, right? T-independent, meaning that the T cells are not going to be needed for us to produce this particular class of antibody. And it's limited, right? It gets stuck in your blood, right? Doesn't travel out of your blood. But it's a good first protection. IgG is our most dominant in our circulation and the longest lived antibodies. They stay around in your blood for the longest amount of time. And it's also the only antibody that can cross the placenta. The bad news with this is this is the antibody that causes hemolytic disease of the newborn, right? Where you don't have that antigen match with the red blood cells and it could cross the placenta and hurt the fetus. So we'll talk about that in more detail later. But that's an important thing to keep in mind for this particular antibody, right? That it does actually cross the placenta.
Is that only during the second pregnancy?
No, it happens when you have an Rh negative mother
That's what
who's actually produced antibodies. And you produce antibodies when you've been exposed. So that's that important thing, right? We talked about this first exposure. So, you know, it could be your first child and you could have this reaction if you've been exposed to positive blood, right? If someone gave you the wrong blood transfusion or you had a miscarriage or something like that previously. Right. Yeah. Yeah. And so we'll review a little bit later. It's also found in the colostrum. Anyone tell me what the colostrum is? It's the first breast milk, right? And so it's very different consistency than the rest of the breast milk that we produce. And these protective abilities of breast milk is one of the main reasons why nowadays people are breastfeeding more and encouraged to do so, because we can't replicate this. What the mother does, and that first milk she produces, she chocks it full of her IgGs. So anything she's been vaccinated against and that she's been exposed to, she makes those IgGs to give to her baby. That's the first gift you give, right? And that is called passive immunity, because the baby didn't make these antibodies. The baby won't be able to do that on their own. Once these antibodies degrade, they're going to have to get their own vaccinations or their own exposures to generate these antibodies for themselves. But it is a short-term protection. And the baby's digestive system, when it's first born, is meant to be able to take up those antibodies and not digest them so that they get that protection. And then the next one we talk about, right, notice it's in breast milk all the time. So all the time you're breastfeeding, you're producing IgA in this dimer form. And it's help coating that baby's mucous membranes, again, against what you've been exposed to, right, until they start producing their own, right? So it helps protect them. And this is not something we can replicate, right? We can't make it in a lab, right? Only humans make it, or animals in general. So one I skipped over for IgG is abbreviated here, but which is bad. I don't know why I don't change that, and I apologize. I have it written out on a future slide, so I'm going to jump to that one. It's this particular protective outcome. It's antibody dependent, so obviously we're talking about antibodies, right? And only this class of antibody can do this, the IgGs. because only that class, the natural killer cells, have an FC receptor for. And so what is happening here is the natural killer cell is recognizing the antibody attached to the cell. Cellular referring to it's a cell that's coated with antibodies. Cytotoxicity referring to, just as the name implies, what is this natural killer cell going to do to this infected cell? Kill it, right? It's the best name ever. They naturally kill, right? But for these guys, especially for this process, they need the help of an antibody, and specifically, it's just IgG that does this. So then I'm going to flip back, go back to my IgAs. So IgAs is the most abundantly produced, but not inside your body, right? We're continually secreting this. How does it help us? Remember, very important for infection to happen, whether it be a virus or a bacteria, they must first attach to us, stick, right? And our mucus helps them not stick, right? We hopefully trap them in the mucus and move them to where we want to go. Well, this aids in that, stopping them from attaching, right? So when the antibody is bound to them, can they attach to us? No, right? Because we're coating them in antibodies. So we're coating them in antibodies, we're trapping in the mucus, and hopefully we're pushing them along their way, and they don't attach, right, and gain entry into our bodies, where they can cause harm. So IgD, as I said, we don't know much about it, very small amount in circulation. It's an antibody that we see as the immune response matures, right? So later in infection. It is a monomer, right? So that single Y we're used to seeing. IgE, again, very barely detectable in blood, unless you happen to be an allergic individual like myself, right? That produces way too much of this stuff. And the other reason why you'll tend not to see it is that when it's produced, it usually is binding to the surface of mast cells and basophils, right? And it can stay there for weeks at a time attached, which is bad news for those of us experience allergy, because the effector cells, right, that have the histamine already have the antibodies on it. We don't have to wait for the antibodies to be found by the mast cells. They already are coated in antibody, right? And remember, it's that FC portion, and in this case, for that specific class of antibody, IgE, we're binding to. Got caught up in my G's. And I don't know why I repeated this slide. It slipped past. So our main antibody classes that are really helpful for us are IgAs, IgGs, and IgM. And remember, IgM is that mega big pentamer when we secrete it, and IgA is that dimer that's going to be in our secretions. All of these guys can do this first outcome of what we call neutralization. Because viruses and toxins must attach to us to gain entry. Otherwise, they can't cause any harm. Okay? So if we have antibodies against the virus and against the toxin, they coat them and stop them from being able to attach, and therefore they cannot gain entry. Right? So we have a special term for viruses and toxins coated with antibodies. We can neutralize them. The next one is somewhat similar, right? Where if we're binding to them, the antibodies are binding to them, they can't attach to us, right? So we can prevent adherence. But in this case, we're kind of stopping, we can potentially stop infection sometimes with bacteria. We can stop them from adhering to us and stop infection. Then the other thing is if they're mobile, say bacteria, right, have flagella. Those flagella move like a propeller does on a boat. If you wrap it up in antibodies, you're going to tangle up that propeller and it can't move. Then you're doing what's referred to as immobilization, right? So, putting down the bayou, right? All of a sudden we stop. What's going on? Pull up the propeller. There's a sweatshirt wrapped around it, right? It was in the bayou. So we literally had to cut it off, really happened to me, off the propeller, so us to continue down the bayou. They can't cut these guys off, though, right? These antibodies bind. They wrap them up, they immobilize them, right? And so that really helps protect us, right? Having the antibodies have this ability. And then the next one is cross-linking, right? So notice, because at the very least, if it's in its monomer form, which is what IgG is, we'll look at IgM in a minute, because that has more than one antigen binding site, right? It has 10. So at the very least, they could grab onto 2 antigens at the same time. It's showing some bacteria here. Outside the body, there are specific types of interactions that happen between antibodies and antigens when you have this cross-linking effect that have specific terminology to them. So you may have heard of glutination. This is when you have a large, unsoluble antigen like red blood cells, right? When we're typing red blood cells outside the body, and of course this can happen inside your body, which you don't want to happen if you get transfused with the wrong type of blood. We take your red blood cells, which we don't know what antigens are on the surface, and we have antibodies. We know exactly what they bind to, what antigens, right? So if we add the anti-A antibody to some red blood cells, and you see this clumping, this agglutination that happens, and you can see this with the naked eye. You don't need a microscope, right? Those of you guys in A&P have probably already done this, right? With fake stuff. But same effect, right? It's going to clump together. You can even do this with bacteria. Usually you're going to need the aid of a microscope just to really see that clumping, though, right? Because they're much tinier than red blood cells, right? So that's agglutination. If you have soluble antigens like proteins, where you can't see them in a solution, right? You can see red blood cells in a solution, right? They're not soluble, they're cells, okay? When you add antibodies, you could cause them to what's called precipitate out of solution, because it's going to form a much larger molecular mass, right? You've got antibodies and antigens binding with each other, and they'll literally fall out of solution, right? You'll see them precipitate out, these large collections now of antigen and antibody. And that's referred to as precipitation, because it literally will do that. It will fall out of a liquid solution. So the next one is what I talked about earlier, right? So again, this is very unique for the Fc receptor for IgG, right? For this process to happen, antibody-dependent cellular cytotoxicity, you need an IgG-coated cell, and you need a natural killer cell to come along, recognize that IgG, and destroy that cell, right? That whole process is referred to as antibody dependent, because you've got to have that antibody. Cellular, because it's going to be a cell that's coated in antibody. Cytotoxicity, because that natural killer cell is going to kill that cell. And then you have opsonization, right? And this is also IgG. And that is because your phagocytic cells, whether it be a macrophage, or who is it that only comes into the area when they're called there during inflammation, a phagocyte? It's one of our phils. Neutrophils, right? Macrophages, remember, are always there, but more are going to come, right? Dendritic cells may come. All these guys have receptors on them for that Fc portion. They have an Fc receptor for IgG. And so they'll grab onto it, just as you see here, and they're going to be able to pull it in. Now, when we talked about complement, right, there was one protein that also is an opsin and will bind, and when it binds, it's going to activate the alternative pathway of complement. What was that one? It's one of the 3 you need to memorize. C3b, right? And I always remember B for binding, right? Tagging for destruction. So same thing, when C3b binds, it's an opsonate. Our phagocytic cells have a receptor for it. We saw this in phagocytosis, right? Grabs onto it, and it's going to engulf and destroy them. Now, complement itself can be activated if there are antibodies bound to the surface of an antigen like this bacteria here. The complement proteins can bind to that Fc portion, right, of the antibody, and it can activate it, and we refer to that as what pathway? It's the classical, right? And there's only 2 classes of antibody that can interact with complement proteins, and they are IgG and IgM. Those are the only 2 that can activate the classical pathway. They're the only ones that can interact with complement. And then once we have complement activated, you'll produce more C3b for opsonizing. So notice, y'all, it was right on the slide all to begin with. Right there. C3b, right? It's an opsonate, tagging for destruction. And then we have inflammation, right? Complement can activate inflammation, which is the movement of the white blood cells into the area, like the neutrophils. There's 2 molecules of complement, letters and numbers you need to know, are specifically involved in inflammation. They are C3A and C5A. And then we could also have the formation, right, of a complex, commonly abbreviated MAC, that could lyse the cells. What's the M stand for? So it's really effective against gram-negative bacteria as opposed to gram-positive, because the M stands for, what do gram-negatives have as their outermost covering? A membrane attack complex, right? So very important, right? This complex of proteins, you don't need to memorize the numbers and letters associated with it, but understand this MAC complex, this membrane attack complex, is inserting donut-shaped protein complexes and lysing those cells. But it has to insert into a membrane. Right? So what benefit do you think this would have, this cross-linking, y'all?
Is it in one area?
Yeah, kind of traps them, right? Kind of nets them in. So again, notice that IgM is really good at this, right? Because it has so many antigen binding sites. This is some red blood cells, right? And again, this is the antibody you produce against the ABO blood group, right? And that's why you get the really good clumping that will happen, because it can bind to a lot of red blood cells at one time and really clump them up. And in our bodies, right, that would help localize them in an area until the phagocytic cells can come and gobble them up, right, and eliminate them from the system. So remember we talked about phagocytosis and how they can escape sometimes, right? And how they can cause C3b of complement not to bind and therefore not become activated by the classic alternative pathway. So, and I think it's the Streptococcus priogens that causes strep throat that has this particular trick up its sleeve. Some bacteria out there have Fc receptors for our antibodies. So when they get into our bodies, they can attach to the Fc portion of our antibodies and coat themselves in this orientation. So they basically just coated themselves in our proteins. Can our immune system see them? No, right? Because our immune system is used to the antigen binding sites being bound to the antigen, right? And that Fc portion sticking out, that Fc region, so that we can grab onto it, right? Or complement can bind to it. In this orientation, we can't get at it, right? And it's going to be even harder to get at and see this bacteria or whatever it is because it's coated in our own proteins. So, although pretty awesome, antibodies are awesome, right? Pretty cool abilities. They're one of my favorite things. We still get sick, right? There's still bad guys out there that have found different ways around our immune system, and even in this case, use our own immune system basically against us, right? Gobble up our antibodies and coat themselves in an orientation in which we don't recognize, right? So the next time we're going to really go into that specificity of, how these cells become activated, right? When they interact with each other, the antigen presenting cells interacting with the T cells and the B cells interacting with the helper T cells, right? That right down to the molecular interaction, right? And how this process goes to the point where we produce these beautiful molecules, right? Of the different classes and different abilities. But it takes time for this to happen, right? And that's the real downfall of this system, is that weak to 2 weeks sometimes it may take before you produce these antibodies, right? Your innate immune response has got to keep these guys under check, keep them trapped in until you can really mount a really specific response against them. Okay? All right. So don't forget